So what I want to do this evening is take us well into the circulatory system, all right? And we're going to talk about the muscular pump and the system of vessels that supply all the tissues. We're going to do it in the opposite order. We're going to talk about the vessels first, and then we'll come back and talk about the heart, all right? And you have to think right away, what are we really trying to do? You've looked at liver, you've looked at kidneys, you've looked at digestive tract, you've looked at lungs. And as complex organisms, you need to tie all those things together. So the oxygen from the lungs gets to all those other tissues, right? So the nutrients from the digestive tract go to the liver where they can be processed and then go out to the rest of the system. So you need a very efficient distribution and removal system, right? And if we look at the evolution of these systems, fishes have a two-chambered heart. One atrium to receive blood, a ventricle to pump the blood. It passes through their gills, and from their gills it goes through all of the rest of their musculature before it comes back to the heart. All right? It's a very fairly inefficient system, but if you live in water and you're not real active, it seems to work pretty well. All right? We go to frogs on land. Being on land takes a lot of energy. All right? And all of a sudden you have a three-chambered heart. And there's a good reason for it, but I won't go into it right now, except that you realize that 3 is bigger than 2. All right? All right, a three-chambered heart for amphibians. And then we see in mammals like yourselves a 4-chambered heart, which is a heart that is characteristic of warm-blooded creatures. Birds also have a 4-chambered heart. Some of you know that because you dissected the turkey heart that you pulled out of the body cavity of your turkey on Thanksgiving. Yeah, I hope you did. So that's a 4-chambered heart. And the 4-chambered heart is a heart that's going to allow very high pressure, all right, very rapid and efficient distribution of all of these components of oxygen and pulling CO2 out of the tissues and delivering nutrients to the tissues and all of the other functions that we want to use this distribution and removal system for. Okay? So as we begin to look at it then and we begin to sort of lay out our vocabulary, we'll start with the heart and let's get all these labels out here. There we go. All right, so we start with the heart. You notice the 4 chambers of the heart. We'll come back to them a little bit later, but I want you to recognize that the heart is subdivided into a right half and a left half. And, of course, you're looking at it in an anatomical perspective there, so the right is over on that side. All right, and you'll notice the right half is blue, all right, indicating low oxygen, venous blood, all right, and the left half is red, highly oxygenated blood. And so what we have is a system that receives blood from the tissues, which is low in oxygen because the tissues have consumed that oxygen. All right, that blood comes into the right side of the heart and is pumped out to the lungs. So we have the right side of the heart responsible for supplying blood to the lungs. All right, of course, that blood becomes oxygenated, returns to the left side of the heart, and the left side is responsible for pumping it out to the body now that it's been oxygenated, all right? And so you have two pumps working in parallel, working simultaneously, right? The right side receiving blood from the body and pumping it out to the lungs, and the left side receiving blood from the lungs and pumping it out to the body. All right? 2 pumps side by side working in parallel. All right? Working together. All right? And if we trace the blood vessels that leave the heart, whether we're talking about from the right side as they go from the right side of the heart to the lungs and through the capillaries of the lungs and back to the left side of the heart, or if we look at the arteries that are taking blood away from the left side of the heart, out to the tissues, and back to the right side of the heart, okay? In either case, the circuit is heart, arteries, arterioles, capillaries, venules, and veins. All right? So that's the sequence of vascular components. And you can be looking at the pulmonary circulation, that is the blood that's pumped to the lungs for oxygenation. We call that the pulmonary circulation. Or we can be looking at the systemic circulation, the blood coming from the left side of the heart and going out to all of your tissues, up to your head, out to your arms, down through your torso and your abdominal cavity and your pelvic cavity, and then down into your lower extremities. All right. And in either case, the circuit is arteries, arterioles, capillaries, venules, and veins. And in the color coding here, I'm indicating here this would be blood coming out of the left side of the heart. It's red. It's high in oxygen. High in oxygen through the arterioles. It goes out to the capillaries, and that's where it gives up its oxygen, right? Because the capillaries are that's the business end of the circulatory system. That's where exchange is taking place, all right? Everything else, heart, arteries, arterioles, venules, veins, all those others are just supporting the events that are occurring at the capillaries, which is where exchange is taking place. Gives up its oxygen, so now we go to blue blood here, venules, veins, back to the heart. Okay? So what I want us to do is to begin to look at these vascular components and distinguish them from one another because they are different in structure, and you'll see that functionally they also are different. In other words, arteries, their job is to carry blood away from the heart. Another way to say that is arteries receive blood directly from the heart, which means that the blood carried in arteries is under high pressure. Structurally, they're going to be different from the veins, where the blood pressure in the veins is in single digits. All right? So blood pressure coming out of the heart on the left side, 120 millimeters mercury. All right? If you cut an artery, you might even hit the roof. All right? If you cut a big enough artery. All right? But if you cut a vein, it's just going to drip out. All right? 1 or 2, 3 millimeters mercury. All right? Versus 120. Okay? So you have blood vessels that are receiving the blood under very, very different pressure conditions. All right? And we'll define other differences between them. Okay, so again, arteries carrying blood away from the heart. And I say usually high in O2 because I'm talking about blood coming from the left side. And the only exception to that is, of course, this artery, the pulmonary artery, carrying the deoxygenated blood from the right side of the heart to the lungs. It's an artery, but it's carrying blood that's low in oxygen. It's sending it to the lungs to be charged up, right? So we can say arteries carry blood away from the heart, usually high in oxygen. And likewise, veins carry blood back to the heart, okay, usually low in oxygen. And so that you don't have to do the high or low or whatever, forget about the amount of oxygen. Just look at it as arteries carry blood away from the heart, veins return blood to the heart. And don't even worry about what its oxygen content is. It's unimportant at the moment, anyway. All right? And so here you see the components then. You have arteries taking blood out away from the heart, subdividing eventually into arterioles, and those arterioles eventually giving rise to capillary beds. All right? That's where exchange is going to take place in the capillaries, and eventually that blood collects into smaller veins called venules, and those drain into larger veins that return the blood back to the heart. So there's your circuit, but you're seeing it sort of in a different rendition here. Arteries carrying blood away from the heart, out to the tissues, subdividing into arterioles, subdividing into tiny little capillaries, reconverging into venules, and then into larger veins, and back to the heart we go. And if you look at these figures here, you begin to appreciate the differences between arteries and veins. And these are worth taking a good look at, you guys. So let's spend a minute here, all right? This is an artery. Here's the lumen. That's the hollow space in the artery. And you can see how thick the wall of the artery is. All right? This is a vein. What do you see there? a really big lumen, and relatively speaking, the wall is pretty thin, isn't it? All right? Look at this one here, and you have to stare at it for a second. This is the vein, this sort of funny-looking collapsed structure here. That's the vein, all right? Here's the artery. So arteries retain their round shape, don't they? Their cylindrical shape, if you will, whereas veins tend to collapse down, all right? If there's blood in it, it won't be quite so collapsed as that, all right? And likewise here, there's the lumen of a vein, here's the lumen of an artery. You can see the wall of the vein is very thin. You can see the wall of the artery here is quite a bit thicker, all right? So with that, let's get into the structure of these blood vessels. And just as in the past we've defined a common sort of basic pattern of structure, we'll do the same here, all right? When we look at the structure of blood vessels, we can see three tunics, all right? So another sort of parallel vocabulary, right? When we looked at the digestive tract, you had four layers, right? Four tunic, the mucosa and the submucosa and the muscularis and the serosal layer. Well, here's what we have in arteries and in veins, all right? You have an internal tunic, the tunica intima, a middle layer, the tunica media, and an outer layer, the tunica externa. And you're going to see that these vary in terms of their composition and their thickness when we go from arteries to veins, all right? So let's just say a little bit about these for the moment, all right? And so the tunica intima, that's the lining, that's the inner tunic, and it is one cell thick. It's one cell thick, and we refer to that simple squamous lining of blood vessels as an endothelium, right? That's the endothelium. It's one cell thick, simple squamous epithelium, okay? There's a little areolar connective tissue underneath it, okay? And in arteries and arterioles and venules and veins, that will be surrounded by the tunica media. In capillaries, that's all you have, all right? Capillaries are just an intima. There is no media, there is no externa, because capillaries are just a cylinder made up of that simple squamous layer of endothelial cells. That's what a capillary is. Tiny, delicate, all right? I've heard estimates that you've got 60,000 miles of capillaries. I don't know if that's true, all right? But you've got a lot of capillaries, all right? Every cell is in contact with a capillary, all right? And the diameter of capillaries is smaller than that of a red blood cell, all right? Red blood cells are 7.5 micrometers across, all right? And a capillary is about 4 micrometers across, which means the red blood cell has to deform itself, and it's going to have a very tight fit as it passes through that, and that's going to allow for really efficient exchange, all right? So capillaries, again, the business end, they just have the intima, that is, they're just an endothelial layer, all right? The tunica media is going to be composed of smooth muscle and some connective tissue, all right? And we'll see it's elastic connective tissue, and it varies in its amount, okay? And then finally, the outer layer, the tunica externa, is the adventitia. What do we mean by adventitia? It's connective tissue that's tying it into the surrounding tissues, all right? That's tying it into the fascia or the connective tissues of the organs that it's passing through, or the spaces that it's passing through. So it's connective tissue, it's elastic to some degree, it's inelastic in other parts, all right? And as I say, the adventitia then is anchoring it into the surrounding tissues. And before we leave this then, a few things. So here's a typical artery in cross-section, and you can see that the wall is pretty thick. What's the thickest layer here in an artery? The media, the tunica media, right on, the one that has the muscle. You're right. The tunica media is the thickest layer. And here in a vein, what's the thickest layer? The externa, right? The media is pretty thin, all right? And it's the externa, all right? So you don't have the advantage of what those muscles do in the arteries, which is to do what? What do muscles do? They change the diameter of that blood vessel, right? And so by changing the diameter of that blood vessel, you are increasing or decreasing the volume of blood going to tissues, and you are also, as you're going to see, able to regulate blood pressure. All right? So you have, sort of two reasons for controlling that smooth muscle tissue. One is to control how the blood is distributed, right? If you're running, where do you want the blood? You want it down here, right? So you want these blood vessels to open up some and give you more blood in those tissues that are active, all right? So the arteries, with their big tunica media, have that ability to change diameter, whereas the veins don't. All right, and we'll say more about these. The other thing I wanted to show you then, is this figure here, which is a classic figure. It's an excellent one to look at. I've already alluded to it. This is the blood pressure as we move from the aorta to the big arteries, the distributing arteries, to smaller muscular arteries, to arterioles, to eventually the capillaries, and then to venules and veins and back into the vena cava, which takes the blood back to the heart. And look at that, you guys. As the heart is pumping, all right, the blood pressure is varying between the moments when the heart is contracting, the systolic blood pressure of around 120, and those in-between moments when the heart is relaxing. But the pressure is still pretty high, right? It's in that vicinity, 80 or 90, right? And so we're just going to take an average here, and we're going to say that as blood leaves the heart, the average of the systolic and diastolic pressure here, the average of this we'll just say is 100. All right? It's 100 millimeters mercury as it leaves the heart into the aorta, all right, and into the large distributing arteries, all right? And it's sustaining, it's holding its pressure, isn't it, initially? Right? It's holding its pressure. It's going down a little bit. I know what you're seeing, all right? But it's pretty much holding its pressure until we get to what? Where does it lose its pressure? At the arterioles. That's right, at the arterioles. The arterioles are where the most friction is taking place, all right? And the capillaries, of course, as well. So at the arterioles and capillaries, you're losing your pressure head, all right? And by the time we get into the venules, we're down here below 20. And by the time we get into that is the veins and the vena cava, we're now down to what? Single digits, right? It's 2 or 3, all right? So if I ask you, why is your blood pressure 120? You now know the answer. Because if it were anything less than that, it wouldn't make it back. It's pumping as hard as it needs to so that that blood travels, and by the time it gets into those veins and into the vena cava, how much pressure is left? Very little. Very little. It would just barely spill over into the heart, all right? It's just very, and that's it. That's why the blood pressure is 120, because that's how hard the heart has to pump to get it to go through the circuit and complete a round trip, all right? Why isn't it higher than 120? You understand now why it isn't lower. It isn't lower than 120 because it wouldn't complete the circuit, right? As it is, it's just barely creeping back in, right? Like somebody guilty sneaking in the back door, all right? Just barely making it back, all right? Why isn't it more than 120? That would be a waste, right? Why do you need it to have pressure when it arrives back at the heart, right? If you're a healthy person at least, okay? All right? So that 120 is set by the fact that you want it to travel through the whole circuit and complete the circuit, meaning have enough pressure to get back to the heart, but no more than is necessary, right? So that you're down to single digits when we get down here into the veins, all right, into the vena cava. All right, so let's take a look at the arteries. And as with anything, we can't just say all arteries are alike. Instead, we're going to think about big arteries, and then we're going to work our way away from the heart. You think about, the further distance you get away from the heart, whether you're going down the body or whether you're going out your upper extremity, for example. All right. As blood leaves the heart, we just said it's under high pressure. And so we have elastic arteries, all right? Arteries that in the thickest layer, which is the tunica media, you alternate a layer of muscle and a layer of elastic connective tissue, and a layer of muscle and a layer of elastic connective. It looks like a Christmas candy, stripy, a layer of red and a layer of white, and a layer of red and a layer of white. Red smooth muscle and a layer of elastic connective tissue, and red smooth muscle, and multiple layers, tens of layers, really thin, all right? And the elastic connective tissue is meant to do what? It's going to expand to absorb the shock of systole, of the heart in its contracting cycle, of the ventricle contracting. It's going to expand to absorb the shock of systole, and then the elastic connective tissue is going to do what?
Store the energy.
It's going to, you could say store the energy. It's going to recoil and keep the blood moving when the heart is in its non-contracting state, all right, when the heart is filling, all right? So it does two things. If we talk about the pumping, ventricle pumping, that's systole, and the ventricle filling and relax in its relaxed moment, that's diastole. We can say that the elastic connective tissue absorbs the shock of systole and maintains blood flow during diastole. Okay? It keeps the blood moving, all right, during diastole. But you're absorbing the shock of systole, all right? And that's due to that elastic connective tissue, okay? When you have hardening of the arteries, then, when those big vessels start to get harder and sometimes even brittle, all right, you can imagine then that the blood pressure downstream is high because you're not absorbing that shock any longer, right? And so that pulse is transferred much further, right? And that can be damaging. And of course, if it becomes brittle, you can have a dissection. A dissection is simply that one of those big vessels just splits open. All right? And when one of those big vessels splits open, you've got minutes, right? You've got minutes, all right? I have a friend. He's in his 70s now. And they live nearby me. And his wife went off to her painting class or something one day, and he felt kind of funny. And it was not funny. He felt like something was really going on. And so he picked up the phone and didn't call his wife. He just called 911 and said, "Come immediately. I'll be laying on the front porch." And he got himself to the front porch, and he laid down, which was smart, right? He laid down so that he kept his system circulating as easily as it could be, all right? Ambulance came, hauled him off to Chomp. Gal at Chomp did some quick analysis, said, "You've got a dissected aorta." It was splitting open. It was leaking out, all right? He had minutes. They zoomed him out to Salinas, cracked him open, saved his life. He's still hobbling around, all right? But it was like absolutely, he knew what to do. The doctor at Chomp figured it out really fast, and they got him quickly onto the table as fast as they could. And it was just like boom, boom, boom. And, he always talks about his miracle, which was really a neat sequence of events. All right. So that's elastic arteries. As we move downstream from the elastic arteries, all right, the elastic connective tissue that was intervening between the smooth muscle instead arranges itself just inside the endothelial layer and just on the outside of the smooth muscle layer. So we end up with what's called the internal elastic lamina and the external elastic lamina, meaning the muscle layer is sandwiched between elastic connective tissue. All right? The blood vessel has different properties. It's still elastic, all right? It's still able to absorb shock, but now we're going to put the emphasis on the muscle part of it instead of on the elastic connective tissue part, all right? And the muscle is going to allow you to change diameter, and in doing so, change the amount of blood that's being distributed to that part of the body. Send more to my arm, send less to my arm, more to my legs, all right? And changing those diameters for distribution. All right? We're not down at the cell level now. We're not saying which cells really need it. We're just saying which region of the body needs it. All right? And so that's what those muscular arteries are doing for us. And then we get down to the arterioles, where there's pretty much no elastic connective tissue in there. Instead, the media is around 6 or 10 even layers of smooth muscle. So they're pretty delicate, all right, the arterioles. And this is where you regulate blood pressure, all right? If your blood pressure is low, shrink down those arterioles, vasoconstrict, and you raise blood pressure back up again. If your blood pressure is too high, vasodilate, all right? And since we're on it right now, you've learned almost always that those two parts of the autonomic nervous system work antagonistically, right? Parasympathetic does one, sympathetic does the other. Not here. All right? One of the Dr. D rules, as I always call it. Sympathetic has full control of vasomotor activity. All right? Sympathetic has full control of vasomotor activity. All right? So sympathetic vasodilates, sympathetic vasoconstricts. All right? And it's just changing the amount of sympathetic that allows you to do one or the other. Parasympathetic, sympathetic, total control, Christina. All right? Of vasomotor activity. Vasomotor, which means both vasoconstriction and vasodilation. Both. All right? All right? There's only one place I told you where parasympathetic increases blood flow. And that's erectile tissue, as you remember. Remember point and shoot? Right? Point, parasympathetic, right? But that's the only case, all right? Otherwise, it's throughout the rest of the body, which is everything that really counts, when you're thinking about distributing the blood out to the system. Forget about the other for the moment. All right? It's all about sympathetic, having that control. Okay? All right. When we get down to the level of capillaries, so we've gone from arteries in their various forms to arterioles and now to capillaries. We can define different capillary types, and I've been telling you about these throughout the course. So these terms are definitely not new to you, right? And so what do we know? Capillaries are an endothelium with a little bit of areolar connective tissue around them, all right? And how many liters of blood do you have? 5. All right? You've got 5 liters of blood. Okay? And if I were to measure the volume of all your capillaries, you have 7 liters of capillaries. Huh? That doesn't work, does it? Because that would mean there's no blood left for the arteries or the veins or the chambers of the heart, right? Turns out that the capillaries, that all those 7 liters of capillaries, only 5% of them get blood at any one moment in time. All right? So blood flow through the capillaries is intermittent. All right? Blood flow through the capillaries is intermittent. In fact, if you measured of your 5 liters of blood that you have, only 250 mLs is in the capillaries at any single moment. All right? Only 250 mLs, right, in the capillaries at any moment. So let's look at these different capillary types. All right? Continuous capillary. What does that mean? That means the endothelial cells are fitting together just like a jigsaw puzzle that you built correctly, where all the pieces are just fitting really nicely together, right? That nice jigsaw puzzle. That's a continuous capillary. It's the kind of capillary that you see in the skin and in muscle tissue, in lungs, where you don't want to see fluid leaking out, right? I don't want pulmonary edema, right? So it's the most, what am I looking for? Continuous? You can't use the word continuous to define continuous, all right? Secure. That's the most secure, all right? It's the non-leakiest, right, of the capillary blood types, or capillary types, all right? And so exchange of materials is going to occur through little clefts between those puzzle pieces, between the endothelial cells, all right, or by diffusion across those endothelial cells, or by pinocytosis, by the transfer of little vesicles of material, all right, between one side of the capillary and the other. All right, so when we talked about the respiratory barrier, all right, remember the respiratory barrier, type 1 pulmonary epithelial cells, right? And then we said there was the capillary next door going through the endothelial cells for diffusion. That's it, continuous capillaries, all right? Fenestrated, I told you about fenestrated. I said take a piece of Swiss cheese and roll it into a cylinder. That's your fenestrated capillaries, little tiny holes in it, all right? And that's going to give you a lot of transfer of materials, right, between the what's in the capillary and the surrounding tissues, or vice versa. And so this is what you find in endocrine glands, is my favorite example, because you have some hormone-producing gland, right? And you want those hormones to get into the blood, right? So how are they going to get into the blood? They're going to pass in through those little openings, through those fenestrations, those little windows, those fenestrations, okay? All right, where else do you see that? The kidney, right? Part of the filtration barrier. Remember the filtration barrier of the glomerulus was made up of the podocytes, right? The podocytes with their little pedicel spaces, and the continuous capillaries with their fenestrations, okay? Or the fenestrated capillaries with their little windows, all right? I didn't know this till today. The intestinal villi, which makes total sense. Remember the villus and that little capillary that goes in and out of each of the villi, and it has the little lacteal going up the middle? Remember that? That little bed of capillaries there, that's also a fenestrated capillary, which I didn't know, right? And then finally, the leakiest are sinusoidal, all right? And sinusoidal, where do we find those? In the liver, where you have a huge transfer of materials between the blood and the hepatocytes, the liver cells, right? Very, very leaky, and it's leaky because there is no basement, or no basement membrane there, all right? And so those cells have huge spaces in between them. There's a lot of transfer of materials between the sinusoidal capillaries, right, that are running in from the, what are the corners called? The corners of the lobules, from the portal triads, right on. The blood running from those portal triads into the middle of each lobule, called the central vein. Very good, all right? And you have a lot of exchange of materials there, all right? Permeable to cells even, and large proteins, all right? And your liver is like that. It has sinusoidal capillaries, and your spleen likewise has sinusoidal capillaries. And you can imagine the same thing. In your spleen, you're trying to do what? You want blood that's going through. What does your spleen do? Yeah, it's going to scarf up aging red blood cells, but it's also one of your lymphoid tissues. So it's checking the blood for antigens, right, for foreign things. And so you want foreign things, which in many instances are bacteria, right, or big proteins, right? You want those to be able to be accessible to all the white blood cells that are clustered around the outside of the capillaries, right? So you have sinusoidal capillaries there for a lot of exchange, okay? All right, so again, back to this figure, all right, is to recognize, what did we say? Blood flow through the capillaries is intermittent. It's not flowing through a capillary all the time. It's intermittent because, as I said, only 5%, any given capillary only has blood in it 5% of the time. All right? All right, so you're opening and closing them and opening and closing them, all right, continuously. All right? And that flow of blood through the capillaries is controlled by small little rings of muscle that sit at the entry of each little capillary bed. So here you have one of those little rings of muscle. It's called a pre-capillary sphincter. All right? And what are those pre-capillary sphincters doing? They're doing this. Right? They're doing this. They're opening and closing on the order of 5 to 10 cycles per minute. Boop, open. Wait 10 seconds. Boop, open again. So blood passes intermittently, all right? And that blood is going to serve that particular group of cells, right? Those tissues there. Enjoy that for a bit. I'll be back later, right? Open, let a little blood in, close, live off of that oxygen, live off of those nutrients, right? Allow waste to get back into the blood, and then open again. Flush that out, bring in some new, right? So it's intermittent, all right, by virtue of these pre-capillary sphincters. And the pre-capillary sphincters are actually responding to the local tissue environment. So they will relax and open if there's high CO2 levels, all right? Or if the pH is going down, which is the same as saying a high CO2 level, right? If it's becoming acidic in the tissues, that'll open them up as well, all right? And so they're opening and closing in response to local tissue conditions, all right? Now, you know that sometimes you can direct blood around the capillary bed, right? Like when you blush or not, right? You're not blushing right now, as far as I can tell, all right? And so you are sending that blood, all right, through one of these divergent channels here, all right? Here we have a thoroughfare channel, which is basically the channel that allows you to direct blood through various capillaries, all right? Or if you don't want blood going through that tissue at all, you use one of these, what's called an arteriovenous, right? Artery, venous anastomosis, meaning you're just a detour around that capillary bed, all right? There is no reason to send blood through that capillary bed. Okay, so the point being, you have multiple options for controlling the amount of blood that's passing through a tissue. Okay. All right. We've taken the blood under high pressure through the arteries, and we've seen that succession of different artery sizes. We've then gone into the arterioles, where we could control their diameter, and into the capillary beds. Where's our blood pressure at right now? It's low, right? We're getting down into the 20s. Go ahead, Edie.
What controls the blood going through the arteriovenous?
It's going to be sphincters. It's going to be.
And then we have hormones, or?
Those are going to be neurally controlled. Yeah, and all of these sphincters that are of larger sorts are going to be neural control, as well as local conditions. Okay. A combination of those. All right, so veins, all right. Now, blood pressure, we just said, is low, isn't it? All right, and that blood has to get back to the heart. So we will refer to venules, really tiny veins, all right, and veins as a low resistance pathway back to the heart, all right, because the pressure is low, all right. Now let's get an image in our heads here, all right? If I have a bucket of water, all right, I got a 5-gallon bucket here, my Home Depot bucket, all right, full of water, and if I empty it, right, I cast it across the floor here. Tell me what that water is going to do? What are you going to see happening here? It's going to spread out all the way to the wall and wash up the wall. What's going to happen? It's going to spread out and slow down and stop, isn't it? Why did it stop? Because water, liquid, running against a surface has friction. All right? It's experiencing friction. All right? And so, likewise with the blood. As the blood is flowing through the blood vessels, there's friction between the blood and the walls of the blood vessels. All right? And the only way then to make sure it gets back is to minimize the resistance. All right? And so the veins are a low resistance pathway, all right, for blood to return back to the heart. And one of the things that gives them their low resistance is that they are not elastic. They're not squeezing in. As you saw, their cross-sectional appearance was kind of shaggy, wasn't it? It was irregular, wasn't it? All right? Veins are not elastic. That nice round, ready to, extend and recoil. They are compliant. All right? Compliant. You get the difference? Let me remind you, just so you get the sense of what compliant is. Your bladder, when it's super full, is compliant. And if it was elastic, you get it? You'd have a powerful stream, right? It isn't. Your bladder is compliant, all right? It's just a little low level of recoil, all right? Well, likewise, the veins. The veins are compliant, all right? And when we look at their structure, again, we've said it's the thickest part is the tunica externa. It's not elastic connective tissue out there. It's collagen fibers, all right? It's inelastic. It's collagen fibers, all right? The other thing that the veins do, well, we'll come back to this in a second. So another point then. The blood is flowing slowly through the veins, all right? And to prevent it from backflowing, the veins have valves in them, okay? The veins have valves. And you all have an assignment when you go home, all right? I want you to find some valves, all right? So there's one, all right? Let's see, it's right there, all right? Here we go, all right? So there it is, all right? Watch. See how the vessel's poking out? Yeah, because there's a valve right there. All right, and what I'm doing is I'm pushing the blood backwards, and it's pushing up against that valve, and the valve is keeping it from backflowing through the further downstream. All right, so you go home and you push some blood backwards, or if you don't want to do that, go to Safeway. And just look at the lady standing at the cash register. Right back there, all right, where that blood is sitting on those valves, all right, and the veins are expanding. Those are varicosities. And what's happened there is what? There's valves all the way up the veins, all right, all the way up. But if a valve collapses, if a valve breaks, then the blood from that would have been held by that valve is now sitting a couple of valves down, right? And if that one breaks, it's sitting there. And what's happening to the vein when you have all this blood sitting on that valve? The vein is distending, right? That's your varicosity. That's your varicose vein, all right? And how do we treat that? We strip out those veins, all right? Ultimately, if there's a concern about it, you strip them out. Because your veins, unlike your arteries, are highly variable. All right, there's a few veins that you can give names to, and they're consistent throughout our anatomy. But most of them, you look at my veins here, get them to stand out a little bit, and you know what? If you look at yours, you got a different layout than I do. In fact, I think if I compared my right with my left, they might be a little different. All right, because veins, their job is just to do what? Just to get the blood back, right? It isn't like it better go this way to make sure these tissues get what they need, and that's what arteries are. Arteries have a very precise layout, right? But veins, not necessary. Just bring it home, right? Any way you can.
Can you just get veins from a different part of the body and then put it in there for like a surgery for that?
For varicose veins? Well, you've got a redundancy of veins, don't you? And you saw when I pressed here that you've got this one converging on that valve, and so is this one. So I can strip this one out, and the blood will just take a different route, right? So you've got a redundancy. And in fact, there isn't like a genetic blueprint for telling you how your veins should be laid out. All right? Likewise, there isn't for most of your smaller arteries. Your genetic blueprint just says, grow out some blood vessels, enough to be able to drain the tissue. And that's how it works. All right? There isn't a plan, all right, for that. It's just make sure there's enough to bring it home. All right? Now, if we take the 5 liters of blood, all right, if we take the 5 liters of blood and we think about where is it situated in these components, the chambers of the heart, the arteries, the arterioles, the capillaries. We've said 5% of it's in the capillaries, all right? I haven't given you percents for the other part, all right? The venules, the veins, 60% of your blood volume is in your veins, all right? 60% of your blood volume is in your veins. And so when we think about your veins, they're like a blood storage reservoir. And if I constrict them a little bit, what am I doing? I'm displacing that blood to other parts of the circuit, right? If I say, well, now the veins only have 40% of the blood, that means that the remaining 60% is spread out throughout the other tissues, right? So when I'm exercising and I've got a bunch of arterioles wide open and a bunch of capillaries saying, me, all right, I can decrease the volume of those veins a little bit and displace that blood so it's able to fill those other tissues and perfuse those other tissues adequately. All right, does that make sense? Now, blood flow returning in the veins then is under very low pressure. And to prevent it from going backwards, we said the veins are equipped with valves. Likewise, your lymphatic vessels, all right? Lymphatic vessels are vessels that are bringing back tissue fluid that's leaked out of the vascular system and bringing it back towards the heart. They are built like veins as well. They have valves. All right? But that doesn't bring it up all that well. All right? You're still relying on the blood pressure. You're still relying on the heart to push it along. So you have a couple of systems besides the valves that prevent backflow to encourage venous return. All right? One of them is simply the action of your muscles. So here you see the muscles in the calf here, and as those muscles contract, the bellies get wider and they push against the walls of the vein. And if you push against the walls of the vein, knowing it has valves, what's that going to do? That's going to force that blood back up, right? Because the valves don't let it go down. So every time your muscles contract, okay, you're doing what? You're putting pressure on the walls of the blood vessels, the veins that pass through there, and we call that the musculoskeletal pump, all right? Or your textbook calls it the skeletal muscle pump, all right? Either way, all right? So activity encourages venous return, right? If you're in the military and you're standing at attention, lock your knees. Flex your legs, just barely. So your pant leg, so your crease is still nice and straight on your pants, right? But your knees are bent just a tiny bit. So your muscles are working to hold you up so you don't, all right, you're not locking your knees. If you lock your knees, you got no pressure on the muscles, and you stand at attention and you pass out, all right? You pass out. So you want to keep those muscles working, all right? Skeletal muscle increasing venous return. All right. Next time you're down here tying your shoe, and you stand up quickly. Ooh, so hold on. All right. What can I do? What can I do to stop that dizziness? I was seeing stars there for a minute. Not fast. There's an easier way. How can I fix that?
Sit down and tie your shoe.
Huh? There. Done. Contract those abdominal muscles. What are they going to do? They're going to squeeze on the veins. Boom! Blood goes right up to the heart. Out of the heart into my head. Boom, done. All right? You try it. You stand up and you feel that way. You feel that dizziness. Next time you're picking something up, done. That's it. That's it. It's done like that. Just like that. All right? It's done just like the musculoskeletal pump. The other thing that increases venous return is every time, listen, every time you inhale, what happens to, you guys already know this, what happens to pressure in your thoracic cavity when you're inhaling? Is pressure increased or decreased? When you're inhaling, pressure is decreasing, right? You're lowering the pressure in the thoracic cavity, right? If you had a straw here in your drink, all right, how would you get the liquid to come up the straw? You would create a vacuum in your mouth, right? Listen. You could draw the liquid up by sucking on the end of that straw. Trust me on this. You could also draw the liquid up by sucking on the side of the straw, if the straw was sort of flexible. Because by sucking on the side of the straw is going to expand, right? Which is creating a vacuum that's drawing that fluid up. All right? Are we good there? Every time you inhale, the side of the straw. Yeah. All right? Every time you inhale, what are you doing? You're sucking on the side of the straw being those big veins coming back into the chest cavity, right? I have a prediction. In 5 minutes, you're all going to stand up to leave. And when you stand up, every one of you is going to be inhaling. I got it. Every single one of you will be inhaling. I guarantee it. And you've been doing that all your life, and you've never realized it. Not one of you exhales when you stand up. Why is that? Because when you stand up, you don't want to leave the blood down in your seat and down in your legs. So what do? That's right. Everybody. As soon as you stand up, you're creating a vacuum and you're drawing the blood. You watch. You watch. You'll see. All right? And those guys that faint when you stand up, I know you're just messing with me. All right? Okay, so let's say a little bit about the heart here, you guys, and we'll just say a little bit about it, and we'll come back on Tuesday and wrap this up. Okay, so of course the heart is what? It's the driving force for moving that fluid right through the system, for moving that blood from the arteries and out to the arterioles and through those important capillaries, the working part of the blood of the vascular system, all right? And as we've said then, the heart is 2 pumps in parallel, all right? On the right side, the pulmonary side, it's pumping under lower pressure, all right? At its peak, about 40. Why? Why is the pulmonary side only pumping 40? Only has to go here, all right? Boom, done, back, all right? That was easy, all right? Whereas the left side has to pump it up to here and down to my toes and out to my fingertips, all right? And so its pressure is about 3 times, all right, the pressure on the right side, all right? And you're going to see that the muscle on the left side is about 3 times thicker than the muscle on the right side. In fact, you could even see it in this little drawing here, right? Here's the heart muscle on the right side. Here's the heart muscle on the left side. Look how fat that is, because it's going to generate 3 times the pressure, all right? We're going to see that the chambers of the heart are separated by valves, so you don't get backflow, right? Anytime you hear valves are to prevent backflow, or the proper term for that is regurgitation, all right? Backflow, all right? And the contractile activity of the heart is intrinsic, all right? What do I mean by that? That means if I cut all the nerves going to your heart, snip, snip, it keeps beating, all right? It's intrinsic. The heart muscle cells beat on their own at about 90 beats per minute, all right? 90 beats, that would be its intrinsic rate, all right? And now you rely on parasympathetic to apply the brakes, because right now your heartbeat is lower, 70, all right? And so you're applying the brakes. Sympathetic is saying intrinsic rate is 90, slowing it down to 70, all right? And when you get up, all right, and you're ready, you're walking to your car, the heart's going to beat faster, so you're going to remove the brake pedal, a little less sympathetic or a little less parasympathetic the brake pedal, right? That's what I just said. Didn't I say that? Yeah. Did I say sympathetic?
No, you said it.
Thank you. Parasympathetic, slowing it down. Sympathetic. So you're going to remove parasympathetic, remove the brake pedal. It's going to speed up as it approaches 90. If you want it to go higher because you're running to your car, then you're going to apply some sympathetic to speed it up even further. All right? So your autonomic nervous system can regulate heart rate, but heart rate is intrinsic. And you can even take heart muscle cells and grow them in a petri dish, and they do their little thing right there in the petri dish. All right? All the time. Close the door of the incubator, they're still in there doing their thing. All right? Because that's their nature. All right? All right. Your heart sits in the thoracic cavity, in the central part of the thoracic cavity that we call the mediastinum. All right? It's upside down. It's an upside down triangle. So if we look at the heart here, all right, this is the base of our triangle, and this is the apex of our triangle. So it's an upside down triangle. The great vessels, the aorta, the pulmonary artery, the great vessels emerge from the base of the heart. All right, so you have to be clear in your mind when you say the base that you're not talking about the lowest part. Rather, you're talking about the uppermost part. It's an upside down triangle. all right? And it sits in a sac, all right, in the pericardial sac, and the pericardium is made up of, this is the last thing I'll tell you tonight, it's made up of a dense irregular connective tissue covering, all right? That's the fibrous pericardium, all right? And that surrounds the space in which you find the heart, all right? It's attached around the body of the heart, all right? Right near the top of the organ, okay, where the great vessels, and then it comes down over the heart and splays out onto your diaphragm, all right? So that's the little cavity in which the heart is going to sit. Now, when your heart forms, you remember your heart forms up here, doesn't it? Right? Remember that nerve? Remember that one? Anybody remember the name of it? The laryngeal recurrent nerve, the nerve that controls your vocal cords. All right? Sneaks through when the heart is still up here in your throat. All right? And then the heart drops down, and it carries that nerve that sort of threaded itself through in your early development. All right? Kicking and screaming down into the thoracic cavity. And why is your heart up here when it forms? Because that's where it is in fish. Good for you. There you go. All right. And so, when it drops down, there's a serous membrane there. And it drops down into that serous membrane. All right. And here, think of a balloon. And now the heart drops down into that balloon. And so now what do I have? 2 layers of serous membrane. Right? Think of a balloon. I'm going to push my fist down into that balloon. Now I have 2 layers. All right. The one that's opposing the surface of the heart is the visceral layer, all right? The visceral layer of the serous pericardium, all right? And the one that's against that fibrous layer, that's the parietal layer of the serous pericardium, all right? And in between those two is the pericardial cavity filled with serous fluid so the heart can move independent, all right? And that's all I'll say with one more thing. If blood leaks into the pericardial cavity, it's going to prevent the heart from being able to fill all the way. All right? That's called Tempanade, if anybody's ever heard of that. All right? It's going to prevent the heart so the fibrous pericardium actually sort of sets the limit. The heart can expand and fill up to that level. All right? It sort of contains the total volume that the heart can fill. All right? But if you put blood into that pericardial cavity, now you're reducing the amount of space that the heart has to expand as it fills up, right? And it's not going to be able to work well. So if you have a dissection of a blood vessel or a coronary artery that has ruptured, and you get blood in that space, all right, you can see it's not going to allow the heart to fill, which means you're not going to pump the blood you need to. Okay, so we'll stop there. Go ahead, inhale.